Hi guys, Kenna here. Today we're going to go ahead and talk about the cell transport. We're going to introduce you to the basic concept of diffusion, talk a little bit about what we call the concentration gradient with respect to membrane, and then we'll kind of look at the difference between passive and active transport. So let's begin by looking at our essential questions for today. So, number one, what is diffusion? You probably heard the term, although you may or may not know exactly what it means. Today, we'll try and give you a very clear definition of what that is so you can better understand how it functions. Number two, what are the factors that impact the rate of diffusion? So, diffusion can go quickly or slowly depending on a whole bunch of different factors. We're going to highlight some of the key ones here. And then number three, what is the difference between passive and active transport? And if you know the words passive and active, this may be pretty clear to you already. All right, so let's remind ourselves of the definition from last class of homeostasis. Pause, see if you can come up with its definition, and then we'll move on. So homeostasis is the process of maintaining balance or equilibrium. And this is incredibly important for cells. They have to keep the proper concentration of nutrients and water coming in while at the same time eliminating wastes. And so the main processes by which they do these tasks are listed here. Number one, diffusion, which I mentioned will be the focus of today, as well as facilitated diffusion. And we need to identify what separates those two out. And the next class, we'll go ahead and focus more on active transport and osmosis. So, what is cell transport? Well, at the simplest level, it's simply moving something from one side of the membrane outside the cell to the inside of the cell on the other side of the membrane. And the simplest form of transport is known as diffusion. So let's define diffusion right here. You'll probably want to get this in your notes. Number one, diffusion is the movement of particles from an area of high concentration to an area of low concentration. Now, when we start talking about this, this is a key point to make sure you get. So again, area of high concentration to an area of low concentration. Okay? Make sure you get something along those lines in your notes. This is going to be a key feature here. High to low. All right? Okay. So, it's always going to keep moving until it reaches equilibrium. And there is no difference between one side of the membrane and the other. So we're going from the higher area to the lower area of concentration. But I also want you to realize that it doesn't just stop at that point. The molecules have some inherent energy and they're going to constantly keep moving. This is what's known as a dynamic equilibrium. Particles will move freely and are evenly distributed. Okay? So, even if we reach equilibrium, so there's a balance on both sides, there are particles that are still moving. And so from time to time, it may go out of equilibrium for a short period of time before moving back to equilibrium once again. Now, most of you have experienced diffusion before. If you ever walk into a grocery store that has a bakery, you've already discovered diffusion. Now, when they have the air circulation systems, they like to put the intake valve right above the ovens, okay? And that's because then it goes ahead and pumps the smell of the fresh baked bread throughout the store. So when you walk in the store and you get that quick hit of, that smells like fresh baked bread, that's on purpose, okay? If you've ever walked down the hallway at school and smelled perfume or cologne, okay, this is another example of diffusion as well. Okay, so diffusion is a passive process. You don't need to put energy into the system. It's the natural movement of the molecules that causes them to disperse and to spread out. So if you watch this diagram here, you'll notice that we go ahead and begin. Let's clear our drawings from before. You'll notice that we go ahead and begin with all of our molecules right down in here in the corner. Okay. And then as time goes on, they move around, they run into other particles, and they begin to spread out. Okay. So you can see one over here, you can see one over here, and I'm drawing straight arrows, but in reality, they don't follow that path. Okay. So let's clear this again. Let's try and reset this. Okay. Oops, too far. Okay, so if you watch these, okay, you can see that it's gone. Let's see if we can move it some more. Okay, now it's over there. Now it moves up here. So it keeps going as it goes ahead and bumps into one particle. It goes ahead and continues to move. And as it does that, the blue particles begin to get mixed through the red liquid that it's in. Whether it's gas, whether it's a fluid, you can actually do this yourself in the kitchen. If you want, go ahead and get 3 clear microwave safe glasses. Go ahead and put some water into them. And when you go ahead and do this, put one of them in the freezer, keep one of them on the counter, and then go ahead and microwave one of them for, let's say a minute or two. Okay. So if you set the one in the freezer for a while, and then you go ahead and pull it out and then do your microwave for the 2 minutes, you can go ahead and put, say, 2 to 3 drops of food coloring in each one of them. When you do this, you'll notice that the particles that are being dropped into the cold one, they go ahead and go in and they tend to just sit there. They don't really move very much. Now, if you left it for long enough, they're still going to diffuse through. The temperature of the water is going to increase. The movement of the molecules will continue to happen, but it's a pretty slow process. In the room temperature one, it'll go at a pretty reasonable pace. And in the one that you microwaved, it's going to go ahead and go through the liquid pretty quickly. It's going to disperse and spread. Now, when we go ahead and start looking at this process, um, you're not really adding energy to the system. You're just using the natural kinetic energy that's in the molecules, because remember, temperature is really a measure of kinetic energy. Okay, so the higher the temperature, the faster those molecules are moving, which is why diffusion occurs more quickly in hot materials, hot gas, hot liquid. Okay, and this makes sense that it has to be some sort of fluid, whether it's gas or liquid, to move, because solids just vibrate. They don't actually flow, and so you can't get a spreading of the material. So I've created a diagram in Paint to help kind of paint this picture for you. I realize it's not maybe the best or most dynamic thing, but I think it gets the idea across. So we start here with inside of the cell and outside of the cell divided by a cell membrane. Now, we've gone over this term for the cell membrane. Does anybody remember what it is? That's right. It's semi or selectively permeable, meaning it allows some things through and others to not go through. Now, you can probably guess just from my diagram here which ones are going to go through and which ones are not. So if we go ahead and start counting up how many of each of these there are, we can go ahead and say 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So we have, let's choose a different color here. We have 11 reds, okay? And we've got 1, 2, 3, 4, 5, 6 of the larger ones, okay? 6 large. Now, as time goes on, there's far more on the left side over here and far less on the right side over here. So the molecules are going to want to move over here to the right side. However, the larger molecules can't fit through the gaps you see here in the membrane, and so they don't. So what you end up seeing is the red molecules begin to go ahead and move from one side of the membrane to the other side of the membrane. And they're going to keep moving until they go ahead and get to equilibrium. Okay. Now you'll notice when we count these up now, so if we go ahead and clear your drawings, 1, 2, 3, 4, 5. We've got 5 reds on this side, and 1, 2, 3, 4, 5, 6. We've got 6 reds on this side. Are those numbers balanced? No, but they're really close. And so what's going to probably happen over time is you'll have 5 here and 6 here. Then it'll switch. You'll have 5 here and 6 over here. But it's going to maintain a ratio that's really close to the same. Okay. So this is what we got to kind of keep an eye on. There are a lot of different things that are going to determine how fast the process I just showed you happens. Let's go ahead and take a look at these. So the first one is the steepness of the concentration gradient. The bigger the difference in quantities on both sides of the membrane, the faster it's going to move. And this should make some sense because the more there is, the more likely it is to move to a place where there's less. Okay. So the steepness gradient of the concentration gradient is the first one. Number two, as we just mentioned a little while ago, is temperature. Okay. The hotter the temperature, the more movement there is in the molecules. This higher level of kinetic energy is going to cause a greater spread of the material. So molecules move faster, so therefore diffusion is faster. Number three, surface area. The greatest surface area, the faster the diffusion can take place because there's more space for the molecules to move through. This is important because we have to start thinking about some items moving through proteins and others being able to go through the actual surface of the membrane. Okay. Now let's imagine for a second that you're a pitcher in baseball. When you're going to throw that pitch, and I'm, let's say, winding up to go ahead and throw the ball against the door at my house, is it easier for me to hit the door handle or just the door itself? Hopefully you recognize that it's easier to hit the door itself than the actual handle because the handle is a lot smaller. It's the same sort of thing when we're looking at the movement of particles. The particle has a greater surface it can hit to go through. It's more likely to go through than if it has a small place to hit and go through. And the last factor I'm going to identify today is the type of molecule or ion diffusing. The bigger the molecule, the more slowly it's going to move through. Likewise, the more charged the molecule, the slower it's going to move through. Remember, we talked about a lot of your body being made up of water, but remember that the cell membrane is primarily made up of lipids. And so, water is a polar molecule. It likes to be around the glycerol head of the phospholipid, but the actual inside of the membrane are those fatty acid tails, which are nonpolar. So nonpolar molecules are going to have an easier time getting through than polar molecules will. All right. So let's give you some examples to see if we can get a better idea of how this works. So the first one I want to use is oxygen. Obviously very important. We need it to go ahead and breathe, to go ahead and allow a lot of these chemical reactions to take place in our bodies, especially cellular respiration at the mitochondria. Now, oxygen is a nonpolar molecule. So it's going to go very quickly through the membrane, but it's also really, really small. So it has the best of both worlds and it's going to go through very quickly. Then we look at carbon dioxide. We have to get carbon dioxide out. It's a primary waste when we start looking at things like cellular respiration, but it's polar. And so it struggles a little bit more than oxygen does, but again, because it's so tiny, it's still able to diffuse quickly through the membrane. And then we go ahead and look at water, also polar, just like carbon dioxide, but also very small. So it still goes through the membrane itself. All of my examples that I gave you right here, okay, all of them are going to go through the membrane itself. So they're going to go this way or they're going to go this way, but they do not need the help of a protein. Okay. If it's a transport protein, which we'll talk about here a little later today, it can go through there if there's space, if there's not something else already in there, but it's not limited to that. It can actually go through the membrane, squeezing in between the phospholipids. So for all three of those, that's what we're looking at. Okay. Now, diffusion that we just showed you is a form of what we call passive transport, meaning there is no additional energy needed to move the particles. We're going to talk about an additional type of diffusion called facilitated diffusion, which needs the help of proteins. But I want you to realize that it's still a form of passive transport, meaning that we do not have any additional energy needs. Essentially, the protein then acts like a tunnel that allows the molecule to fall through. It doesn't use an energy assist. We'll talk more about that next class. So, why do we need facilitated diffusion? Well, not all things are as small as oxygen, carbon dioxide, and water, and so some of them can't fit through the membrane itself by going between the phospholipids. So, if we look at things that are larger, like glucose or amino acids, or something that's heavily charged, like a sodium ion or chlorine ion, they can't go through the membrane itself. They need assistance, and they will often go through some sort of transport using a protein. So these molecules pass through protein channels instead, and this process, if it does not require energy, is called facilitated diffusion. So the movement of molecules still passive, just like regular diffusion by definition is. The only difference is they need the help of some sort of protein channel to help it fit through because of its size or charge. All right. So here's an example of a lipid bilayer, our fluid mosaic model we talked about 2 classes ago. And when we go ahead and take a look at this, the one I've circled here is a transport protein. Notice that it's a tunnel, that it's not closed. And so you may have a carbohydrate portion of a glycolipid that's attached to a glycoprotein, and it's going to signal to send certain molecules over here that are going to fit through this channel. And so as they fall through this channel, really do want you to think about it like falling, okay? Because they're not being grabbed by that protein. The protein just sits there and the molecule falls through it. So let's go back to my paint drawings for a second so you can kind of see how this goes. If we get back to my same one, but we find a section of it that has a protein channel, notice that the red dots that we were looking at before are already at equilibrium. But now we can go ahead and watch the larger molecules begin to transfer from one side to the other. And they're going to have the same restrictions on rate of diffusion that you saw with the smaller red particles. So over time, diffusion occurs, and those large molecules are going to move across until we reach equilibrium. Now, in this case, unlike before, we have 3 on each side. Does that mean that it stops? The answer is no. Remember, diffusion is a dynamic process. The molecules always continue to move as long as there's kinetic energy in the system, and therefore they do not stop. They will always stay close to this, but for a very short period of time, you may have 4 on one side and 2 on the other again. But that's for a very short time before it goes right back to 3 and 3. Okay. So it wants to stay at equilibrium, but because it's a dynamic system, there is some movement involved. So let's take a look at this idea of cell concentrations. Now, there are 3 super important terms, so please make sure you get these down. The first one is hypertonic solutions. Hyper means above or beyond. So this is going to be more dissolved solute, okay? More. So hyper equals more. Hypertonic, okay? Now, when we start looking at this, if I go ahead and have a membrane and I want to go ahead and identify how much is on each side, let's say we have 7 on this side, and let's say we go ahead and have 4 on this side. The side with 7 is going to be the hypertonic side, okay? 7 would be the hypertonic side. Now, we also have the term hypotonic, okay? When we go ahead and take a look at hypotonic, hypotonic, let's separate that, is less dissolved. So in this same situation, while the 7 side was the hyper, the 4 side is going to be the hypotonic, okay? So we have hyper, and we have hypo. Okay. So the side with more is hypertonic, the side with less is hypotonic. But as we saw in our previous example, once we did the movement of the large molecules, sometimes they're the same. Well, we have a term for that too. And if you guys remember your geometry, for example, or if you're in it now, iso means same. So like isosceles triangle. So iso is the same. So in this instance, we would have, say, 3 on one side and 3 on the other side. There's no hypertonic or hypotonic because the numbers are the same. Okay, so those are the key terms for today, and make sure you know those along with the other key vocabulary that we're going to look at. And we'll go ahead and continue our discussion of cell transport next class when we go ahead and add in osmosis and um, active transport. So today we focus on diffusion and facilitated diffusion. All right, thanks, guys. Hope you're having a good one. Take care of yourselves. Bye bye.